Yeah, awesome. Yeah, welcome everybody to this month JETZON call. JETZON is the joint exploration of the Twilight Zone ocean network, and we do these calls usually one time in the kind of more European time zones and one time this one for more Pacific countries. So yeah, I'm really stoked about our speakers for this month. We start with Connor, who I just met, as we just said, at the last Ocean Twilight Zone symposium in Woods Hole last month. So super cool to see you here. And yeah, if people have some new research to share, just message me or Adrian Martin, who is the JETZON coordinator. If you want to present in one of the next JETZON calls, we're really keen usually to especially highlight ECR research, but also, yeah, usually just normal mesopelagic research. So yeah, Connor, the floor is yours if you want to start.
Sure. Sounds great. Thanks, Venya. It's great to be here chatting with the folks from the JETZON community, and thanks for the invite to come and talk. As you said, my name's Connor Shea. I'm a PhD student at University of Hawaii, and today I get to share, I guess, some highlights from a paper we just had published in Limnology and Oceanography, titled Small Particles and Heterotrophic Protists Support the Mesopelagic Food Web, Zooplankton Food Web at Ocean Station Papa. The work was done as part of exports and the National Science Foundation, or was funded by the National Science Foundation. Let's see. Yeah, so I'm going to give a real brief background on exports and Station Papa, and then introduce sort of our overarching research questions and the methods that we used for this paper, and then get into some results and just highlight sort of one implication of the work. So, as many of us probably know, what the main goal of exports really is to develop our tool set to help basically estimate global CO2 uptake from the oceans using satellite ocean color data. And it was basically broken into two main field components, the first of which was at Ocean Station Papa in the late summer of 2018. Station Papa is a HNLC region. It's iron limited, and so it's characterized by relatively modest rates of primary production throughout much of the summer. And the food web in the euphotic zone is really efficient with a fairly low transfer efficiency, just meaning that a relatively small proportion of that NPP is actually going to make it into the mesopelagic zone. One of the key questions highlighted in the exports program is this question of what are the main controls on the efficiency of the vertical transfer of organic matter below the well-lit surface ocean, right? So in the mesopelagic zone. And so that's where our work is really focusing, basically on that question. Our piece of that project is basically asking, how does the structure of the mesopelagic zooplankton community affect the functioning and the efficiency really of the biological pump? So for this paper, we were really focused on the zooplankton food web down in the mesopelagic, which you can sort of see situated. Can you see my mouse if it's on this screen, or do I need to be over here? You see it now?
I can see it
Okay,
now.
cool.
Yeah.
Good, thank you. So, we're really focused on this community in the mesopelagic zone and trying to understand, like, what are the relationships with these different arrows I have in my little food web diagram, which represent sort of movement of carbon throughout the system. So for this paper, we're really focused on two specific goals. The first is to determine the major sources of organic matter to this mesopelagic zone community. And this is supposed to help us better understand the biogeochemical roles of zooplankton within the community, right? To what extent are they really attenuating flux, or are they repackaging small particles into larger, faster sinking fecal pellets? What are they really doing down there? And then two, to determine the trophic structure of that zooplankton food web. And the idea here is basically to help us understand how efficiently is this community processing material and what proportion of that material gets sort of shunted into the dissolved organic matter pool. And then the thing I want to highlight at the end today basically will be, how do these qualities vary regionally? And I want to highlight this because it sort of pertains to one of the themes that came up a lot at the JETZON workshop last month, where we're trying to understand what are the bulk qualities of the mesopelagic community in these different sort of characteristic oceanographic provinces, and how do we understand that? Okay, so now to get into some of the methods. I wasn't actually at Sea in 2018, so I've got a lot of kudos to give to the team that was out there collecting the samples that I used for this work. So we collected a bunch of particle samples by in situ filtration. Here's a bunch of the team members that were doing that. We collected multiple sizes on different size filters. And then the isotopic composition of those samples was actually determined by Paul Wajtel and Hilary Close, our collaborators at the University of Miami. And Paul actually put out a really awesome paper on isotopic composition of those particles, really honing in on the different degradation mechanisms and transformations those particles are undergoing as they sink through the mesopelagic zone. So check that paper out. We also collected a bunch of zooplankton samples by mock nest toes. Debbie and her team and collaborators were responsible for that collection. We collected samples down to 1,000 meters, and for isotopic composition, we separated them into 5 different size classes. So here's a photo of Debbie with the mock nest, I think actually at Station Papa. Okay, and then the last sort of methodological piece I want to share are just like a couple of key concepts for stable isotope analysis, so we can all be on the same page as we go through the results. And so basically, when we're looking at stable isotopes, we're really measuring the ratio of naturally occurring stable isotopes in different sample types. And so we can think about this as an isotope ratio, the ratio of the abundance of the heavy isotope to the lighter isotope. But more conveniently, we refer often to this contrived delta value, which essentially is our conversion of that ratio into a value that's sort of it's got a per mil is the unit, and then the delta value has meaningful variation on the scale of like 1s to 10s of per mil. So we'll talk about delta values a lot today. And we're interested in the nitrogen isotopic composition in particular because it changes as a result of processes where bonds to nitrogen are being formed or broken. So one of these processes that we're really interested in here is that of trophic fractionation or trophic enrichment, where basically we observe that the delta 15N values tend to increase from one trophic level to the next, or these tissues are becoming enriched in the heavier isotope nitrogen 15 as we move up trophic levels, and that's depicted in the schematic from Chikurishi et al. 2014 here, where at each trophic level, right, this delta value tends to be increasing fairly predictably. This is true when we look at bulk nitrogen in tissue samples and also in a subclass of amino acids that we call trophic amino acids. So, right, the poster child for these trophic amino acids really is glutamic acid, and we see a roughly 8 per mil enrichment in glutamic acid at each trophic level. We can also look at a different class of amino acids called source amino acids. And we can think of these as being conceptually similar to essential amino acids. They show relatively little fractionation with each trophic step. And so I'm showing phenylalanine as an example of that here, which means that they're really useful. And we'll see why in a minute, but basically we can think of them as reflecting the isotopic composition at the base of the food web, even when we're looking at them in a higher level consumer, like a zooplankton or a fish or something like that. And then the last thing I want to hit on here is basically that we can basically compare the delta 15N value of trophic and source amino acids to come up with an estimate of trophic position for a certain organism, just from a single sample. And so most commonly, glutamic acid and phenylalanine are used for this calculation. But what we're going to see a little bit later on is that we can use different combinations of trophic and source amino acids to learn about different qualities of the food web. And so what I'm going to show basically is that if we look at an alanine-based trophic position, this should give a picture of the trophic ecology in a food web that includes metazoans, but also heterotrophic protists, right? So if we've got an amoeba zoa consuming some particle, that's going to be reflected in this estimate of trophic position, whereas glutamic acid will not. So by comparing those two estimates of trophic position, we can start to understand what portion of this food web is mediated by metazoans versus protists, which is pretty cool. Okay, so to get into results, we're going to first talk about the sources of organic matter to the food web, and then go into some results on trophic position. What I'm showing you here is a plot of the delta 15N value and source amino acids for particles in the top 500 meters, right? So this is data from Wojtel et al. 2020, or sorry, 2023. And right, the first thing I want to point out basically is that the delta 15N value in large particles in source amino acids stays relatively constant with depth, okay? And then when we start to look at smaller size classes of particles, like the 1 to 6 micron particles shown in green, and the 0.3 to 1 micron particles shown in red, we're seeing an enrichment in the delta, or in the nitrogen 15 and the heavy isotope of nitrogen with depth, an increase in the delta values. And so what that results in is basically a distinct isotopic composition for these different size classes of particles in the mesopelagic zone. The next thing I want to point out is basically that, right, if we look at the isotopic composition of sediment traps, which are shown in these black triangles here, they line up really well with the large particle samples that were collected, indicating that they're doing a really good job at collecting those larger, faster sinking particles in the mesopelagic. Now let's put some zooplankton data on there. So showing the same particle data here, but now I'm showing a bunch of zooplankton samples throughout the water column in these black circles. These are actually a bunch of different size classes that I've just smushed together for this particular plot, or lumped together, but they're all collected during nighttime tows. And then we also have two specific taxa. We have Metridia that was collected at, it's a copepod we collected at 25 meters, so the square up here, and the Neocalinus is this triangle. They're both vertical migrators, but with pretty different migratory behaviors. And right, the first thing we notice is that these zooplankton look pretty different than the sediment traps that we saw a moment ago. And they line up really well with these 1 to 6 micron particle size class, right? And so the initial interpretation of this data is, well, this is a community that must be reliant on small particles, and most likely this 1 to 6 micron size class. But if you think into it a little further, you really realize that we essentially have 4 different possible sources of organic matter in this system. We've got vertically migrating zooplankton, as characterized by Metridia and Neocalinus, which look like particulate in the surface waters. We've got large particles, and then really just various different size classes of particles throughout the water column, likely with different sort of synthetic histories, where these small ones we think are highly microbially degraded by the time they're down in the mesopelagic here. And so these zooplankton, right, their delta 15N values, yeah, they could be relying on a food web based entirely on 1 to 6 micron particles, but you can also produce a zooplankton with isotopic composition like this if it's relying entirely on, right, an equal parts mixture of these submicron 0.3 to 1 particles and the vertically migrators, right? So there are multiple possible solutions that could result in zooplankton that look like this. So what we decided to do was basically put together a pretty simple 4-source isotope mixing model, and then try and use Markov chain Monte Carlo simulation in order to identify what are all the possible solutions to this mixing problem and which ones do we think are the most likely. When you do that, you get a solution space that looks like this. So what I'm showing you here is a ternary diagram. And the way you read off this diagram is basically any point in this ternary space represents some composition of submicron and small particles, large particles, and active particles, which you can read the contributions off of the axes here. And we've lumped together the 2 smallest size classes of particles here, just so we can fit it all on this sort of 3 axes. And what we see is that the solution space, basically for these zooplankton, for this mixing problem, all of the solutions essentially implicate a substantial role for these smallest size classes of particles in supplying organic matter. And the place with the highest solution density is where those particles are the dominant source of organic matter to this food web. So one of the big takeaways here is just that these small and submicron particles have a really important role in supplying organic matter to this mesopelagic zone food web. And then the last thing I just want to come back around and highlight here is basically that these particles weren't efficiently collected in sediment traps. And that doesn't necessarily mean that sediment traps are doing a bad job characterizing sinking flux. They might be doing a great job, but whatever is making its way into the food web down there has at least undergone some sort of process like microbial degradation before it's incorporated by the animals at depth. Okay, next I want to talk about food web structure. So I'm going to kind of actually fly through the first half of these results and then spend a little time summing it up at the end. But basically, so here I'm plotting trophic position based on glutamic acid and phenylalanine for particles shown in these black circles across depths. And the main trend in this data basically is that we just get an increase in trophic position for these particles into the mesopelagic, which indicates that they're either like fecal or microbial in character. They're not really phytoplankton anymore. Next, we see that the trophic position of zooplankton stays relatively constant with depth. And that within those zooplankton, the large animals tend to be about one trophic position higher than the small animals. We can look at an alanine-based trophic position as well, like I talked about earlier. And the main difference we see there is just that zooplankton tend to have higher trophic positions now when we use alanine. And basically all this shows us is that heterotrophic protists have to be an active component of this food web. And in the paper, we really make the case that, you know, we might actually not be that interested in the absolute value of trophic position in this case. We really want to know about what's the extent of the food web at some depth. You know, what's the difference in trophic position essentially between zooplankton and the material making up the base of the food web, because that tells you how many steps are happening at a certain depth. And so we can calculate that here, and that's what I'm going to show you in this plot. And when we do that, we get a relatively different picture of what this food web looks like. We actually have a decreasing food web length as we go from the euphotic zone into the mesopelagic, from about 3.5 steps to 2 steps in the mesopelagic zone. And then we can actually calculate the number of protist and trophic steps implicated in that food web. And we see that it's roughly 1 throughout the entire water column. And what this is telling us is that basically grazing by heterotrophic protists is a pervasive component of this food web all the way down to at least 500 meters. Okay, and so the last thing I want to do here today is highlight, we have a bunch of cool points in the discussion that I encourage you to go read, but I want to highlight one of those, which is this part where we compare our results from Station Papa with results from other locations across the North Pacific Ocean. Basically, amino acid stable isotope analysis was done at Station Papa, at Station Aloha during the summer and in the winter, and then at these 2 equatorial sites that were actually surveyed during an El Nino year when they were relatively low productivity. And in this paper put together by Sonia Romero in 2020, simple 2-source mixing model was used, similar to what I just showed you, to assess the relative importance of small and large particles in organic matter supply to the mesopelagic. And the basic question being asked here was, can we predict organic matter supply dynamics from some basic oceanographic parameters? And what Sonia found, here's a photo of Sonia, is that when flux is low, flux into the mesopelagic in particular is low, and when diel vertical migration is weak, we see that these smaller size classes of particles tend to be really important. And this is evident by zooplankton with relatively high source amino acid delta 15N values. And so then what we did for our paper was basically take the data Sonia compiled and then add what we saw at Station Papa to that compilation. And the results for the mixing model I'm showing here are a little different because we use the same 2-component mixing model Sonia uses, 2-source mixing model, and we're only looking at 1 to 2 millimeter zooplankton, that portion of the community. But what we see basically is our results shown in the left panel here aren't that consistent with this trend Sonia observes, with an increasing importance of small particles at low carbon fluxes. But we do see that it supports this trend where small particles are relatively important when migrant biomass is pretty low, right? And so what this suggests essentially is that at all of these sites, which are all fairly low productivity, small particles do become really important only when active transport becomes fairly weak in that system. And right, so this is kind of showing this interesting reciprocal relationship with regards to organic matter supply, but also starts to show us how we might start to understand or predict this food web structure sort of spatially using some relatively simple oceanographic parameters. And to finish up, I guess I just want to highlight some things I really want you guys to take away. One, sediment trap material may not necessarily represent the actual material that's being utilized by the community in the mesopelagic. Two, heterotrophic protists play an important and active role at the food web at Station P, converting this small microbially degraded POM into a form accessible to mesozooplankton consumers. And then last, at low productivity sites across the North Pacific, there appears to be this reciprocal relationship between diel vertical migration or kind of this microbially degraded POM in organic matter supply. And I guess this last thing is that, right, as our conceptual model continues to grow, we have to start to employ sort of new statistical techniques that accommodate that. And so right now I'm working on applying similar methods to a really different site, the North Atlantic, during a spring bloom, and then developing and testing sort of more sophisticated models to deal with this system. So with that, I'll finish up. These are those two papers I talked about, and I'd love to take any questions you guys have for me today. Thanks for listening.
Awesome. Thank you so much. That was great. Are there any questions? Feel free either to raise your hand or actually, I don't know if I can see it actually. Here we go. I'll just change the speaker view so I can see. Or just write your question in the chat. That would be helpful. Or just start talking. I mean, we're not too many people, so feel free to. In the meantime, you said there will be that the isotopic composition in the material you collected in the sediment traps says that there's more large particles in the trap. Maybe it's a misunderstanding, but do you think it's just undersampling the small particles, or do you think that they're just utilized before they enter the sediment trap, or they're just basically, well, ingested or whatever by the community before they enter the sediment trap?
Sure. Yeah. I think there are a couple different ways you could explain that difference, right? One, maybe you have this really small size class of particles that just due to hydrodynamic effects with the trap, they just don't get collected, you know?
Yeah.
You have sort of maybe some sort of a bow wake in front of the trap, and they're so small and sinking so small, they just are sinking so slowly they just go out of the way. But the alternative explanation would be that you could have large particles sinking down that disaggregate and form this suspension that would never be collected in the first place. And then they undergo these microbial transformations before being consumed by animals at depth. So they're sort of delivered there by those sinking large particles, but then are produced in a sense by disaggregation and consumed once they've undergone some transformation after that. So those are the two that I'm kind of working with, but I'm sure there are other explanations.
Yeah, interesting.
I'd love to hear. And Paul talks a lot about that in his paper on microbial degradation of those particles.
Yeah, interesting. And then you showed that the vertical migration or the migrant biomass was higher in summer than in winter, or actually no, there was a lower. I don't know. Did you? Oh, you did check during one season, right?
Yeah, I'll bring that slide up right now.
Yeah.
Right. So yeah, at Station Aloha, they surveyed it during the winter and during the summer, and migrant biomass, was a lot higher, or 50% higher in the summer than in the winter.
Oh yeah.
And was related to a lesser utilization of small particles during that season. The idea there, I guess, would be that you just have active transport, maybe bringing fresher or just alternative material that can be consumed and incorporated into the food web as opposed to this like, relatively degraded stuff that's maybe been there for a while.
Yeah, okay. Yeah, that makes sense. Any other questions? Don't be shy.
I
Oh,
have one.
Blake, here we go.
Hi, Blake.
Hi, Connor. Great talk. That was awesome. Could you go back to your 10% takeaways slide?
Oh, yeah. Yeah.
Okay, I had a question about the heterotrophic protist part. So heterotrophic protists play an active role in the mesopelagic food web at station papa, converting small microbially degraded POM into a form accessible to mesozooplankton consumers. So I guess that conclusion's from the trophic position calculated from alanine, right? It's like that it's a higher trophic position, so therefore those protists must be an important part of the food web. So
Yeah.
are we assuming at this point that the protists are then taking like the really, really small particles that are maybe getting reworked by bacteria and then feeding on that, and then the protists are then the food source for the zooplankton? That's the conclusion there.
That's the idea. Yeah, that's the idea, exactly.
Okay, and then my question was, are you looking at the 13 carbon essential amino acid values for that as well? Like, I'm just wondering if, because some papers have shown that like the bacterial like signature, I guess, is a little bit different from, let's say, like the primary producers, diatoms, that kind of stuff. And I'm just wondering if the protist then would look like the bacteria if that's what they're feeding on. And I was just wondering if you saw that in the 13C data, if you looked at it at all.
I see what you mean. Yeah, like you have some sort of upgrading of that organic matter by bacteria that makes it more nutritious, which is why you'd have proteins targeting that, and maybe feeding on the bacteria that are growing and not necessarily on the like sort of substrate organic matter. Yeah, I honestly, I've looked at that carbon fingerprints, and I really fully expected to see a solid bacterial signature, but I don't at all. If you do that carbon fingerprinting on this data set, it all looks like microplankton, or sorry, phytoplankton.
Mm.
So that was a surprising result. But it's not published in this paper. I think I'm still figuring out how to deal with that data. Yeah, and
Cool.
I brought that up with some of our co-authors, and I think the explanation given essentially is like, if you have bacteria synthesizing amino acids de novo, then then the carbon isotope should look like this bacterial fingerprint signature. But if they're incorporating amino acids as is from the substrate that they're feeding on, then they might not impart that signature, and you might not see it in the carbon isotope fingerprinting. But yeah, your guess is as good as mine on that one.
Gotcha. Cool. Thanks.
Yeah, thanks, Blake.
Incorporation of the carbon isotope fingerprints of microbes would mean that the protists are actually eating the bacteria that degrade it, and that's not necessarily consistent with this. So it's not inconsistent either, but they're not eating the bacteria. The bacteria are pretty small, but so are those particles. They don't have to be particle attached in order to degrade it.
Mm.
So they could be free living, exuding enzymes, and utilizing what comes out of that. You understand? So
Yeah.
in order for the protists to acquire a carbon isotope fingerprint of bacteria, they have to be eating that bacteria. And they're eating the organic matter that's being degraded by the bacteria. So it's still detrital material, but it may not necessarily have to have bacterial biomass. Right?
Gotcha. Yeah, that makes sense.
I was thinking the same thing that Brian just said, but with another twist on it, is the bacteria that might be on those particles, even though they're small particles, the amount of carbon that is actually a bacteria carbon compared to the amount of carbon in that particle that is particle carbon, it might also just be that the bacteria signature is being flooded out by the particle signature, right? Like there's way more carbon coming from the particle than the bacteria themselves. Like whatever ones aren't, say, free living, right? That was my thought.
Yeah, awesome. That makes sense. Any other questions for Connor? Okay, I think then we move on to our next speaker. If you
Thanks,
could
guys.
unshare your screen. Thank you so much. That was
Yeah.
great.
Bye.